Hello and welcome back to another deep dive. Today we're going to step away from the textbook for a moment and go straight into the kitchen. I want you to picture something very specific. It's morning, you're making breakfast, and you decide to fry an egg.
A classic start to the day.
It is. But think about what actually happens in that pan. You crack the egg and the egg white comes out. It's clear, it's runny, completely liquid. But the second it hits the heat, something really strange happens. It turns opaque, white, and it becomes, well, solid.
Complete transformation, yes.
And it's so weird, isn't it? I mean, usually you heat a solid, like ice, and it becomes a liquid. Here, you heat a liquid and it becomes a solid. It feels like it's breaking a rule of physics or something. Why does that happen?
That is a fantastic question. And that simple act of cooking breakfast is actually the perfect way to get into today's topic. That hardening of the egg white is all about proteins. And that means today we're tackling a really important part of your syllabus. Unit 10, biomolecules.
Biomolecules. I know for a lot of students this chapter can feel a bit dry. Lots of diagrams and structures to memorize.
It can feel that way, but we need to think about the why. Proteins aren't just about, bodybuilding and protein shakes.
That's the first thing that comes to mind, isn't it? Muscles.
It is, but biologically, proteins are the worker molecules inside every single one of your cells. I mean, if you don't understand proteins, you can't explain how antibodies fight off a fever. You can't explain how oxygen gets from your lungs to your feet.
Or even genetic diseases, right?
Yeah.
Like sickle cell anemia.
Exactly. That's a protein problem. So the stakes are very high. We're not just memorizing things for an exam. We're trying to figure out how life itself actually works.
Okay, so let's start at the very beginning. Ground zero. If a student needs to picture a protein, what is the most basic, fundamental thing they should be thinking of?
At the simplest level, you need to think of a polymer.
A polymer.
Yeah.
So like a long chain made of repeating units, like a plastic chain or something.
Yes, but with one huge difference. A polymer, like starch, is made of just one thing, glucose repeated again and again. Proteins are what we call heteropolymers.
Hetero, meaning different.
Precisely. They're made from building blocks called amino acids. And there isn't just one type. There are 20 different types that can be strung together in any order.
Okay, that sounds a bit abstract. I need a visual.
Let's do a mental movie. Imagine you have a long string and a box of beads. But these aren't just any beads. Imagine they are all tiny magnetic beads.
Okay, a necklace of magnetic beads. I can picture that.
Now, in your box, you have 20 different colors of beads. And each color has its own unique personality. In chemistry, this is the R group, or the side chain.
A personality, what do you mean?
I mean, they behave differently. Some are bulky, some are small, some have a positive charge, some are negative. And this is the most important part. Some of them are shy, they hate water, and others are friendly. They love water.
I think I see where this is going. If I string all these different magnetic beads together, they aren't just going to hang there like a limp rope, are they?
Not at all. That is the magic of proteins. Because of all those different charges and personalities, the moment you finish stringing them, the whole thing just snaps together. It twists, it folds, it clumps into a very specific shape.
So the string makes its own shape automatically.
Instantly. And if you change the order, let's say you swap one red bead for a blue one, the whole pattern of how the magnets attract and repel changes. The final shape could be completely different.
So that seems to be the main idea here. The sequence of the beads determines the structure.
And the structure determines the function. If you remember one thing from today, let it be that. Sequence gives you shape. Shape gives you function.
Okay, let's get a bit more technical. We have the beads, the amino acids. What is the actual string holding them together?
The string is a very specific type of chemical bond. It's called a peptide bond.
And this is something we absolutely need to know for the exam, right?
A must-know definition. A peptide bond forms between the carboxyl group, that's the A to COH, of one amino acid, and the amino group, the NHNH2, of the next one.
And when they join, something gets removed.
Yes, a molecule of water is removed. We call it dehydration synthesis. You're left with a strong CN bond, carbon to nitrogen. That's the backbone of the protein. It's very stable.
But wait a second. You said the magnets snapping together make the shape?
Mm-hmm.
Are those the peptide bonds?
No, and this is a classic exam trap. The peptide bond holds the sequence. That's the primary structure. But the folding, the final 3D shape, is held together by all those other weaker invisible forces like hydrogen bonds and interactions between the side chains.
Okay, let's try to separate those ideas. The textbooks always have primary, secondary, tertiary structures. It's confusing.
Let's use an analogy. Think of it like origami.
Paper folding? Right.
Exactly. The flat sheet of paper with the instructions written on it, that is the primary structure. It's just the linear sequence of amino acids, one after another.
Right. Flat paper, primary structure.
Now, you start to make small local folds. Maybe you fold the paper back and forth to make a fan, or you roll one corner into a little spiral. That is the secondary structure. In proteins, these are called the alpha helix, which is the spiral, and the beta pleated sheet, which is the fan.
And what's holding those little spirals and fans in place?
Hydrogen bonds. It's like using tiny bits of tape just between the backbone atoms. It's not about the side chains yet.
Okay, so we have these folded sections. What's tertiary structure then?
Tertiary structure is the final overall 3D shape. It's when you take that whole sheet with its little spirals and fans and you crumple it all up into a finished object, like an origami crane. That's the complete functional shape, and it's determined by all those personalities of the side chains interacting.
I want to focus on that crumpling process. You mentioned Adar's shy beads earlier. That feels really important for how the final ball shape is formed.
It is probably the single most important driving force. Let's switch our mental movie, forget the beads, let's go to a school playground.
Okay, I'm on the playground.
Imagine a very long line of students all holding hands. That hand holding is the peptide bond. It's a strong grip they can't let go.
Got it.
Now let's say the playground suddenly floods with water, because the cytoplasm of a cell is mostly water. Some of these students are hydrophobic, they are introverts, and they are terrified of water. What will they do?
Well, they'll try to get away from the water, but they are stuck holding hands.
Right. So they pull the whole line inwards. They all huddle together in the middle of the group to shield themselves from the water.
Like a huddle in a football game.
Exactly like a huddle. All the hydrophobic, water-hating amino acids bury themselves in the core of the protein. At the same time, the hydrophilic kids, the extroverts, they love the water. So they happily arrange themselves on the outside surface of the ball.
So the folding isn't random at all. The protein is forced to fold this way because of which parts like water and which parts don't.
Precisely. The technical term is hydrophobic collapse. It forces the protein into a very specific, unique 3D shape. And that shape is what lets it do its job.
And because it has a specific shape, it can act like a key for a specific lock or something like that.
Yes. Or a tool with a specific function. A pocket to hold a molecule.
Okay, let's test this with some real-world examples. Let's talk about hemoglobin and collagen.
Perfect contrast.
Hemoglobin is in our blood. Collagen is in our skin and bones. Now, you might think, well, if they're both proteins, both made of amino acids, they should be kind of similar.
Like all cars are cars. But that's not right. Think of it like having a big box of Lego bricks. You have the same set of 20 types of amino acids. If you follow one set of instructions, you build a race car. That's hemoglobin. It's globular round, compact, it dissolves in water, and its job is to move around carrying oxygen.
So it's a transport vehicle.
It is. Now, take the exact same Lego bricks, but follow a different instruction manual, a different amino acid sequence. You build a long, sturdy wall. That's collagen. It's fibrous. It forms these strong, rope-like strands that don't dissolve. Its job is to provide structure.
Wow. So one's a taxi and the other's a building beam.
Yeah.
And the only difference is the order of the amino acids.
That's it. Sequence determines structure. Structure determines function.
So what happens if you mess up the sequence? You know, if you're building the Lego car and you put a square brick where a round one should go, is it really that big a deal?
It is a massive deal. And this brings us to probably the most important medical example for this chapter. Sickle cell anemia.
A genetic disease, yes.
It's the perfect proof of this entire concept. In normal hemoglobin, at one specific spot position number 6 on one of the chains, you're supposed to have an amino acid called glutamic acid.
Okay, glutamic acid, what's its personality?
It's hydrophilic, it loves water, so it's very happy to be on the outside surface of the protein. But in sickle cell anemia, a DNA mutation changes the instruction. Instead of glutamic acid, the body inserts an amino acid called valine.
Just one single swap out of hundreds of them.
Just one. But here's the problem. Valine's personality is the opposite. Valine is hydrophobic. It hates water.
Oh, I see the problem now. You put a shy kid on the outside of the huddle where all the water is.
Exactly. And that valine is so unhappy that it creates a sticky patch on the surface of the hemoglobin molecule. It's desperate to hide from the water, so it tries to stick to other hemoglobin molecules.
So they stop floating around freely and start clumping up.
They clump into long, rigid fibers. And these fibers literally stretch the red blood cell out of its nice, flexible disc shape into a hard, sharp sickle shape,
And those sharp cells get stuck in tiny blood vessels.
causing immense pain and damage. all because one amino acid was in the wrong place. This shows how critical that sequence is. Change one bead, and the whole machine can break.
That is just incredible. The level of precision is mind-boggling. Okay, let's go back to where we started, the egg. Now that we understand all this, what is physically happening in that pan?
We can now use the proper term for it, a word you must use in your exams: denaturation.
Denaturation. Sounds like destruction.
It is. When you add heat, you are adding a huge amount of energy. You are basically shaking the protein molecules violently.
So on our playground, it's like a powerful earthquake.
A perfect analogy. The shaking is so strong that it breaks all the weak bonds, the hydrogen bonds, the side chain interactions that were holding that neat 3D huddle together. The protein unfolds.
The tertiary structure is destroyed.
Correct. But here's the million-dollar exam question. Do the students let go of each other's hands? Do the peptide bonds break?
You said those were strong covalent bonds, so I'm going to say no.
You are absolutely right. The primary structure remains intact. You're not cutting the string of beads. You're just untangling the complicated knot it was tied in.
Then why does the egg get solid? If the proteins are uncoiling, shouldn't it become more liquid?
Think about it. Before, you had millions of tiny, compact, soluble balls floating around. Now you have millions of long, unfolded, sticky strings. What do? They get tangled up with each other. They form a massive, messy, aggregated clump.
Like a giant chaotic knot.
A giant, insoluble knot. That is the solid egg white. And that's why you can't uncook an egg. That process of denaturation is irreversible.
Okay, that makes so much sense. Before we summarize, what about in the lab? There's usually a color test for proteins, right?
Yes, you're thinking of the Biuret test.
How did that one work?
It's very simple. If you have a solution that you think contains protein, you add Biuret reagent. If the solution turns from its normal blue color to a deep violet or purple, protein is present.
And what's it detecting?
It is specifically detecting the peptide bonds. The more peptide bonds there are, the more intense the purple color.
Got it. So purple means protein.
Simple as that.
All right, we've covered a lot of ground. I want to shift into exam mode now and get the absolute clearest full marks definitions for the board exam. The clarity lines.
Let's do it.
First, the main definition. If the question is, what are proteins? What is the perfect answer?
You should write. Proteins are heteropolymers of alpha amino acids linked by peptide bonds. Their specific amino acid sequence determines their unique 3D structure and, consequently, their biological function.
Heteropolymers is the key word there.
Okay.
Next, the levels of structure. How do we define them clearly?
Break it down like this. Primary structure is the linear sequence of amino acids held by peptide bonds. Secondary structure is the local folding, like alpha helices, held by hydrogen bonds in the backbone. Tertiary structure is the overall 3D shape held by side chain interactions. And quaternary structure is when multiple chains assemble to form a single functional unit, like in hemoglobin.
That's a great, clear breakdown. Final one, and it's a big point of confusion, denaturation versus hydrolysis.
Yes, this is critical. Denaturation destroys the secondary and tertiary structures. The shape is lost. But the primary structure, the sequence, is intact. Hydrolysis, on the other hand, is what happens during digestion. It actually breaks the peptide bonds, chopping the protein back into individual amino acids.
So cooking an egg is denaturation.
And digesting that egg is hydrolysis. It's the difference between untying a knot and cutting the rope into pieces. Do not confuse them.
Untying versus cutting. I love that. Okay, let's wrap this all up with a quick summary.
Sure, let's recap. 1, the core idea is that proteins are chains of amino acids where the sequence is everything. 2, the visual to remember is the playground. The shy, hydrophobic amino acids huddle inside, which creates the final shape.
The huddle, got it.
3, the key examples are sickle cell anemia, where one wrong amino acid breaks the machine, and cooking an egg, where heat unfolds the protein, which is denaturation. And finally, the mantra to repeat is,
sequence determines shape, and shape determines function.
You've got it.
That is a fantastic revision pack. Before we go, I want to leave our listeners with one final thought. We've talked about how nature builds these amazing molecular machines. What's the next step for us?
Well, think about this. For thousands of years, we could only study the proteins that nature made. But now, with AI tools like AlphaFold, we are finally understanding the rules of this origami. We're starting to learn how to design completely new proteins from scratch.
Proteins that have never existed in nature.
Exactly. Imagine designing a protein shape to perfectly break down plastic waste in the ocean. Or a protein designed to block a new virus before it can even infect a cell. If you can master the folding, you can, in theory, build a biological tool for almost any problem. We are moving from just reading the instruction manual to actually writing it ourselves.
That is a truly fascinating frontier. From frying an egg to designing world-saving molecules, it seems proteins really do it all. Thanks so much for this deep dive.
My pleasure. Keep visualizing those shapes.
And to our listeners, good luck with your studies. We'll see you in the next deep dive.